A lot of people think when they hear the word cancer or tumor that it's just one thing, a collection of cancer cells. It's actually dozens of different types of cells. Cells that bring in blood supply, endothelial cells, cells that build a supportive matrix or fibroblasts, and then lots and lots of different types of immune cells. And so all of those together, that's what forms the microenvironment. That really affects things like how well a cancer is going to respond to a treatment, how quickly or slowly it's going to grow, and ultimately how well a patient does. Our lab broadly focuses on questions related to human tumor immunology. The basic idea is that our body's immune system is really trained to recognize things that are foreign and to get rid of it. And that includes cancer cells. Cancer cells look very foreign to the body, and so the immune system, if it's doing its job, should be able to get rid of it. But we know that clinically, when cancer forms in an individual, the immune system is not doing its job, that somehow that cancer is hiding from the immune system. And so our lab really seeks to understand how that process happens and how we can fix it, how we can hopefully then get the immune system to readily attack cancer cells. We use an approach called single-cell RNA sequencing. So thousands upon thousands of cells, and actually sequencing their RNA. That helps us to really define, when we do this in larger groups of individuals, what does that look like for tumors that are really responsive to treatment or tumors that are resistant to treatment? What's different in those different microenvironments? What might be going wrong in a resistant tumor? How can we modify it? Then we use a lot of patient-derived model systems to actually treat those tumors in a test tube and actually say, if we block protein X or gene Y, does that actually lead the immune system to function better and kill tumor more effectively? And so we can use an approach to steer that immune system effectively to recognize cancer. That might help really control cancer in a long-term or durable way. And so that's what we've tried to do with a therapeutic cancer vaccine study, where we tried to understand what are the best immune targets that might be present in a kidney cancer. And the reality is that's going to be different for every single patient that walks in the door. For each of those patients, we sequenced each individual's tumor. We found those unique markers or footprints that were the best immune targets, and we made a custom vaccine that would be targeting the immune system against that. The fundamental goal of our lab is really with the patient in the center. And so that's not just learning from the patient, that's really thinking with an eye towards getting that back to benefit more patients in the future. And we have lots of ongoing work that studies that with the goal of ultimately moving that towards therapy. Yale is really a unique place, both on the clinical and translational front, bringing therapies to patients and bringing material back, but also on that basic scientific front. And when you put those together, I think that's where new ideas and innovation happens.